Hello, everyone. Welcome to the Antibody Engineering and Therapeutics Digital Week, brought to you by the organizers of the Antibody Engineering and Therapeutics Conference Series. My name is Brandy Fiddler, and I will be your host for today's session, Accelerated Label-Free Kinetic Screening and Early Single-Domain Antibody Discovery, a comparative analysis with phage display samples from Periplasma and comparison with orthogonal technologies. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session. Let's now begin by introducing our speakers for the session. Libin Cui, founder and CEO of Akeagen, and Indrani Chakraborty, manager of application at Gator Bio. Thank you for joining us today. Now I'll hand it over to you to begin the presentation.
Good morning, everyone. I'm Indrani here at Gator Bio, and today we will be talking with our collaborator, Dr. Libin Cui. He's the CEO and the founder of the company Akeagen. And recently we completed a collaboration where we screened 1,000 plus periplasmic solutions that was obtained from his KM mouse platform. And the topic of today's discussion is accelerated label-free kinetic screening in early single-domain antibody discovery. And we will compare different orthogonal techniques that Libin uses in his lab versus how we used Gator in our lab and how the results compare and contrast. So the overview of our talk is, first it will be Libin who is going to give his brief overview of the company, the platform, how he makes those specific phage display samples. And then we will talk about a little bit about our company, Gator Bio, our core technology. And then we will deep dive into the data assay optimization, data analysis, and things like that, and compare the data that we obtained with several orthogonal techniques and summarize at the end. So I will hand it over to Libin from here.
Hi, everyone. Good morning and good evening. So my name is Libin Cui. I'm the founder and the CEO of Akeagen. So today I'll present our platform, which called KM mouse. It's a single-domain antibody discovery platform based on the genetic modified mouse. And it's a really powerful platform technology to generate next generation single-domain antibody for bispecific or multiple functional biologics. And so in today's talk, and we'll present a case, and we in collaborate with Gator Bio for high throughput, high efficient screening of functional antibody in a short time. So Akeagen, I founded Akeagen company early in 2015. At that time, I started working with KM for single-domain antibody discovery. So I found that there are a few challenges in using the KM, the large animal, to generate single-domain antibodies. So first is the number of the animals we can access. The second is the immunization. It takes longer time, much longer time, and also the immunogen we use needs much more compared to small animals. And the third, because in the KM, not only there are heavy chain antibodies exist in the single animal, but also there are conventional antibody exist in the animal too. So after a short time, I thinking whether we can generate a small animal platform that can replace llama or camel to generate single-domain antibody. So this is so we spent quite a few years to finally develop the camel mouse single domain antibody platform. So we know that the heavy chain RNA antibody was originally discovered in camel by a group of scientists in Belgium. And later there's a company called AB-Links, and they focus on using camel camelase to generate therapeutic antibody based on the heavy chain RNA antibody. They call the VH domain of the heavy chain RNA antibody nanobody. And we know that many therapeutic antibody come from mouse, from hybridoma, and later from humanized mouse. And so in mouse or human, there exists only the conventional antibody, which have both the light chain and the heavy chain. After we design and develop using the genetic modified method, we generate this camel mouse, which called CA mouse, means that mouse that replaces camel. So in the camel mouse, all the AGGs is in the heavy chain antibody format, and there is no conventional antibody. So compared to the camel, so it's more efficient for the screening for immunization. And in the camels, and the basic is that we use the endogenous mouse VH genes to generate the heavy chain antibody instead of using transgene like other platforms. They use human VH genes or other VH genes to generate this heavy chain antibody. So the camel mouse we generate, they are very healthy, and so they can breed normally, have a normal lifespan. And in the camel mouse, which I show here, the right panel, and this is a Western blot of the serum of the camel mouse. So as we see that in the heterozygous, which mimic the camel, there's both the conventional antibody and the heavy chain antibody. So I think in this heterozygous mouse, the heavy chain antibody is a major part of the immunoglobulin ADG. And only minor part is in the format of the heavy chain antibody. However, in the homozygous camel mouse, and there exists only the heavy chain antibodies. There's no conventional antibodies at all. So first question is that once we got those mice, we want to test whether they can give an immune response to the antigen we immunized. So this is the case we immunized with the camel mouse with antigen for different time. So after each immunization, we collect the serum and test the binding to the binding activity to the antigen we used. So we can see that after 3 immunizations, and we can see there's a huge increase of the binding activities in the serum to the antigen. And this is the data from BRI technologies, the Gator technology. And we also want to know whether those heavy chain antibody generate from the camel mouse can bind specifically to the antigen. So in the right panel, this is the Phage analysis data. So the left is the control, which we use the HEK293 cell that did not express the antigen on the surface. So we can see that there's no binding at all. The HEK293 cell that expressed the antigen on the surface, we can see there's a specific binding of the heavy chain antibody to the cell lines. So this slide shows that the camel mouse not only can generate strong immune response, but also the antibody, the heavy chain antibody in the serum of the camel mouse can bind specifically to the antigen. And as I mentioned that we use endogenous mouse VH genes to generate this heavy chain antibody. So this is different from other platform technologies that are available on the market. And I list here, this is based on publications. And those platforms use similar technology. This is the transgenic. They transgenic the human VH gene, different number of VH genes into the mouse or rat, and generate this their platform. So the number of the VH gene in those transgenic mice or rat is limited to 8 or 20 VH genes. However, in the mouse genome, there exists 144 functional VH genes. So we take advantage of those huge number of VH genes in the mouse genome. And so we test whether all those VH genes in the mouse can generate heavy chain only antibodies. So we did next generation sequencing for the VH, all the VH from the mouse. And we can see in the left, on the left panel that all 144 VH genes really can generate single-domain antibodies in the camel mouse. However, the frequency, the user frequency is different, and this is very similar to the conventional antibodies, or conventional the wild-type mouse that have both the heavy chain and light chain, the usage of the heavy chain. So with the camel mouse, now we can make single-domain antibodies using the camel mouse. So the procedure is very standard. We first immunize the camel mouse with antigen. Our next slide, I'll show which type of antigen we can use. And then we collect the spleen or lymph node or bone marrow B cells or plasma cells and make a library. Or we can use hybridoma technology as well. And combined with next generation sequencing, we can have a fully coverage all the repertoire of the immunized VH, the single-domain antibody repertoire. And with the leads, and we can go to the next step for characterizations. So the whole procedure take about 3 to 4 months. And the immunization of the small animal is very flexible. And we can use different antigens, as shown next slide. So in our company, we have tried use different antigens. We tried recombinant antibody proteins, synthesized peptide, and we also tried using the mRNA technology coded with RNP. So that's also very good antigens to give high immune response and titers, and also others like very like particles. And so because we can generate a large number of camel mouse animals for immunization, so we can optimize the immunization protocol to either to save the time or to increase the immunization titer. So after immunization, we usually in our companies, we can generate immune feature display library. We also have the Easter display library technology set up in the company. And right now, we also have partners and use the hybridoma. They generate hundreds of hybridoma after one antibody campaigns. And we're also developing single B cell approach and combine with NGS, and we can generate leads for the target. So today I'll focus on the Phage display library. So why we use Phage display? I think the Phage display is a very powerful technology for single-domain antibody discovery. First, we can generate a very large Phage display libraries to cover the whole repertoire of the immunized mice. And so after panning with the antigens, we can get a very high positive rate. And another benefit, which I think is the major focus of today's talk, is that from the Phage display single clones, we can generate the periplasmic samples, and those periplasmic samples are very easy for next step for biochemistry or by other assays, including the functional assays using Phage, and also especially using the BRI next generation BRI technology that developed by Gator Bio. It's really powerful technology for early discovery. So this is a case study, and the antigen we use here is a fusion protein of cytokine with the FC domain. So actually, there are two cytokines, and they belong to a family. So we call it A and B. So they belong to the same family member, and all the two antigen fused to the FC. So we immunize the camel mouse, and we can see that the mice can have a very strong immune response, and so we collect the spleen and also the lymph nodes, which we show here is very enlarged lymph node and enlarged spleen from the mice that have high titers of the antiserum. Then we generate, we amplify the VH genes from those B cells and make a Phage display libraries. So the libraries can be 10 to the 8 to 10 to 9. So it's a large library that has enough coverage for the whole repertoire. So once we have the library, we'll do the standard Phage display panning. So there are two ways. Either we can coat the ELISA plate with the antigen, and that's a solid panning. And we can also use the magnetic beads, and we coat the magnetic beads with the antigen, and using the beads to do the panning for the positive binder of the phage that expresses the VH on the surface. So from the right panel, we can see that after 2 rounds of panning, we get like 90% of the positive read against the different part of the immunogen, so which is the cytokine or the FC. So either cytokine or the FC, or both the some clones that can bind to both cytokine A and cytokine B, because they belong to the same family and they have a homologue. And this is the ELISA plate, show the picture. And on the right corner, so this is Gator BRI data, show that in the the one from one plate, and those single picked VH clones, that's the periplasmic samples, show different binding activity. Some are very strong. So this is real data, and this is the Gator data. I show that the single clones. We can see that in the right corner, and those are negative controls. There's no binding, so the binding curve are almost 0. And we can see in 1/3 of the clone, and they have show strong binding to the antigen, which is the cytokine. And this part, Indrani will give a detailed presentation. And so combined with the periplasmic samples from the immunized libraries, so we can quickly generate the leads. Here I will present one assay we use, which is a TR FRET. And this is based on the energy transfer. So we have cytokines, we have the receptors, we put them together, and then we have we have the donor and also acceptor for the energy transfer. And if there's antibody, if the periplasmic samples with the VH single antibody can block the interaction of the cytokine to the receptor, and we will see a decrease of signal, which is shown on the right. And the blue signal means there are positive signals, those strong binding, and the pink, the less the number means there's a blocking activity. So from one plate, from a randomly picked 92 samples, we can see that we can have many clones that have show strong blocking activities. So from those blocking activities and the primary screening, and we will pick them and do the sequencing and do the clustering, and also do other cell-based functional assays. So from those assays, we can quickly identify the clusters that have blocking activities. So the last slide showed that people will ask how stable are those mouse VH or mouse single domain antibody antibodies. So actually we express the VH domains in the format of heavy chain antibody, which we fuse the CH domain with the human FC region and express them in CHO cells. And after one step purification, affinity purification, we can get very high purity. And with the HPLC analysis, we can see that above 95% of the VH genes show monomer. So it means those mouse VH genes or mouse single domain antibodies, they are very stable. So in summary, so we develop a single domain antibody mouse platform that can generate single domain antibody in a very with high diversity and high affinity, and also with the Phage display, we can quickly prepare the samples for binding activities. So I'll transfer the presentation to Indrani, and she will present the technologies and the data we use that can help us quickly and efficiently to generate lead for the target. So now I transfer to Indrani.
Yes, thank you, Libin. So just to give you all background for Gator Bio, we are based out in Palo Alto, California. And we have actually been existing from 2018, even though our founders are associated with the BLI technology since 2001. And we call ourselves the next gen BLI company. We serve several biotherapeutic discovery and development companies who are focused on discovering antibodies, peptide small molecules as drug candidates. And also we have unique offerings for the AAV analytics area. And currently we have footprints all over the world. And our users are growing every day in numbers. And we have a lot of high impact journals. And so please follow us on the internet. This being Antibody Digital Week webinar, of course, I know that most of you are aware, but still just to give a little bit of a background. This is a cartoon of our machine, and it is so simple and easy to use that this is probably one of the workhorse in discovery or a hybridoma company or any kind of company where you are trying to screen thousands and thousands of antibodies that you are developing through in silico technologies right now. The instrument is, of course, fluidics free. It's a label-free system. We have easy to use software that is used easily to do the assay setup as well as the data analysis. And just because it does not have any fluidics involved, so you can imagine that the collaboration that we did with Libin, which is screening thousands of periplasmic solution, it's very simple because the instrument will not stop, it will not clog, or it can give you data, right? Because it's very easy to use when you have supernatants, cell lysates, or plasmas. Now, I have the next slide, which is a simple one to just show that how or what is the physics or the biophysics principle behind the biolayer interferometry. How does it read, right? So if you think that this is the biosensor, it is all about the white light that is incidented from the top. We have spectrometers inside the machine. And then it's reflected back from the plate surface. So the difference in the pattern that emerges from this incident light with the refracted light is what we see. And the software, if there is any shift in this pattern, it will plot it against time. So that's why we are doing label-free real-time binding. So when you don't have any event going on in the tip of the surface, of course, there is no change in the signal of this incident versus the refracted light. And then you get a straight line. So it's a baseline. And then when you have some analyte that, or a ligand that actually binds to your surface, what happens is there is a right shift. And that right shift, the difference is plotted in the y-axis as nanometer, and that with corresponding to time. So this shift, which is like increasing the nanometer, can give a variety of information. That can be the amount that is being bound to the surface, so you can quantitate your molecules of interest, or it can give you a affinity on rate constant, that what is the binding constant while it's binding, right? And it doesn't stop here. You take the biosensors to the next well, where you have an analyte in the solution that binds to the ligand that it captured. Then the refractive index pattern changes. So you can see the further binding event happening as well. So this, you can build on layers of information. And then not only you can see that forming complexes and things like that, but you can also see dissociation. So when the dissociation happens, you just take the complex that has formed in a buffer solution and it dissociates. What happens is it goes, the complex has an off rate. So it has a left shift. So this is how the software would try to calculate the on rate and off rate by just simply looking at the nanometer changes while it happens in real time. Now, from Gator, what we offer is a solution package. Of course, it's a combination of the instrument that comes in with easy to use software as well as consumables. And these consumables are biosensors ready to use that is catered toward the application that you want to look for. So for this particular assay development or the collaboration that we did with Libin, we had many samples, right? So basically, if we are looking at application areas that what Gator addresses, for this particular project, we are in a very early discovery phase where Libin, he is obtaining this periplasmic solution through phage display. And he has thousands of them where he wants to characterize these nanobodies that he has expressed, that whether they do bind or show any binding towards any of the antigen or not. And if they bind, can we do a ranking or screen this periplasmic solution based on their ranking, like how strong are they binding towards the target antigen, right? And then on top of that, we can also provide several layers of information, that is epitope binning, like do we have any biosimilar for that particular target, that we know that, on which epitope this particular biosimilar binds. So does all these periplasmic solution, maybe we would want some to be in that particular epitope, or maybe we don't want to be in that epitope. And along with that, we also, for this particular case, like he wanted us to also do receptor binding information. So we can even set up those kind of assays here in order to, get more information using very simple assay setup. So for this particular collaboration, we are just here in the first phase. But as you can see, with Gator, and you can extend the application areas not only for the discovery of biotherapeutics, but small molecule discovery, as I was mentioning, AAV and gene therapy as well, because we have a lot of application that caters to all these areas as well, which is not part of today's talk. Now, for the workflow, what Libin did was he sent us 13 plus 96 well plates. We had given us periplasmic samples for the Akeagen cow mouse that was for this phage discovery campaign against a particular target. And so he screened this against, we are calling it antigen A, but he wanted information that whether these huge amount of samples, they bind to antigen A, antigen B, and antigen AB. So antigen A is a homodimer, AB is a heterodimer. And on top of that, all these samples, 1,000+ samples, can we show him the receptor blocking for them, that how does it work? And then after we find the receptor blocking information, can we do epitope binning against a biosimilar? And can we then summarize it and help him do the lead identification process, in the lead identification process, where he matches his technologies that he has been screening previously, like he showed you the FRET as well as the ELISA, in order to narrow down his lead. So how like Gator comes in and provides more information and helps him make more reasonable choices, basically. So for that, as you can see, the number of samples are so huge. So we had to go for our highest throughput current offering, which is our Gator Pro. So Gator Pro, simultaneously, it's a 32 high frequency channel machine, and it can do 32 kinetic experiment at the same time, and it can hold 396 or 384 well plates. So the throughput is huge, right? And if you are doing quantitation, it takes it does like 1,000+ quantitation in 2 hours. And for our assay set, shows 1 hour for each plate. And so each plate, it's subjected to 5 different experiments. So it will be tested against antigen A, antigen AB, which is a heterodimer, antigen B, and then we will do blocking and receptor epitope binning, and as well as a receptor blocking. So all the assay setup and everything, of course, you can use liquid handler as well. That helps you pipette and makes it even more fully automated. That way you don't have to spend that much hands-on time. Now, then the biosensor, then the question comes, what biosensors do you want to use? So in the very beginning of setting up this kind of a humongous study, we want to make sure we are looking at the cost, of course, and as well as also not only the cost, right, but at the end, you also want to get a data that is really meaningful and adds value. So we decided that so the antigens that we were given had biotin. So we were using SA and SA-XT. And also one of the antigen has the twin Strep-Tactin protein, which is, so we have a Strep-Tactin XT antigen, biosensors, which binds very tightly to this twin Strep-Tactin. So these were the 3 biosensors that we narrowed down, we will use for this collaboration. Now, just for the twin Strep-Tactin, this is very good alternative in case you have this tag. The reason being, this biosensor, you come in, you load your antigen, the binding of this twin Strep-Tactin with the Strep-Tactin XT is so strong, it generates a very stable baseline, thereby allowing you to do very good kinetic experiments. But at the same time, it's regenerable. So it's cost effective and you can regenerate 20+ times. So when you are thinking of running this thousands of samples, this is a very good alternative. That's what we found. Now, what or how did we set up the study, right? So first thing is we had 2 types of study. One is we want to understand the binding event. First, of course, yes or no binding, which we want to compare with ELISA. And then next thing is we want to take it to the next level. We want to provide Libin, our collaborator here, with more information. So this is a periplasmic solution. So for periplasma, we cannot do quantitation. So we cannot do the association constant. So all we can do is we can determine the dissociation constant, which is the off rate. And we can rank these periplasmic solutions based on the off rate. So the stronger the binding, the off rate is tighter, right? So what we do is we set up all the solutions, the antigen, the supernatant, all in a 96 well plate. And we have the program and the automated using our picker. It takes the biosensor from one position to another position. So it goes to baseline, loads the antigen, washes, comes in with the supernatant, which is the association, and we see the dissociation. Right? And then what happens is we come in with this huge set of data, because you can imagine like almost 3,000 plus data just for doing kinetic screening, right? Like how do you analyze the data? So we bring in our software here to funnel through the right kind of information in a way that we do not miss out any good lead for him. So we have added, what we did was we added checkpoints. So the things that we decided that we would add checkpoints is we want to make sure first the assay ran well. So we did the loading height. Loading height is the height of the antigen that binds or that is captured by our biosensor. So if this is not captured well, then of course the assay doesn't make sense to begin with, right? So we will not use that data. That was the first checkpoint. The second checkpoint is response. So we want to see how the periplasma, or what is the amount of response of the periplasma. Now, if the periplasma itself binds very low, right? And what happened in his case, that is each of his plate, because of his platform, he has so many positive leads that we had choices to eliminate or set up a cutoff threshold of a certain nanometer height that below that we would not look. So we'll only take samples that are above that particular height. Then we saw the K-off value, which was fitted by the software. And not only did we check for the K-off value, but we also wanted to understand the goodness of fit, because, you know, we have to go through thousands of values, right? So we might like interpret or misinterpret a data rather. So we took the dissociation R and dissociation R square value, and which is what we used in order to show the data here. So these were all the QC data that we obtained, and these were the checkpoints. Now I'm just showing you here as an example of plate 5. Just one plate we picked, and this is his ELISA selection. And I know the small cartoons here are very tiny. But then our software gives you a value of all the KDs. And when we zoom in just one plate position, you can see that when one supernatant or periplasmic solution, in this case, it's binding, it shows that you have a very nice association followed by a dissociation curve. But when you don't have something binding, you all can see is noise, right? And this matched very well with what he had selected using his ELISA. But when we compare it with ELISA, of course, you know, it's you are getting one data, which is endpoint data. But here we see the entire binding happening in real time. The system is fully automated. And not only we get a yes and no kind of an answer, but what we do is we also get to rank in terms of a real dissociation constant, right? So I can compare many yes and no, but in just for the sake of time, let me just move a little bit quickly. So the next set of experiments that we did is the epitope binning and the blocking. And just to make it simple for the software user and everything, we kept both these two experiments very similar. So what we did is we took our assay probes, load the antigen first, then come in with, when we were doing the receptor blocking, we did the receptor capture first, then followed by the antibodies later. Here you can see, and when we come in with the biosimilar, same thing. So we first bind the biosimilar and then come in with the antibody. And the way we interpret the data is very simple and clear. So if anything that blocks the receptor, of course, when the receptor is bound to that particular antigen, it will be a straight line. It will not show any binding, right? And when it does not show blocking, that is, it can bind to the antigen even when the receptor is bound or the biosimilar is bound, it will show binding. So you clearly in real time, we could see these events happening. So for Libin's Fred assay, he has a number, right? that this is his blank, and then he himself sets cutoff and things like that, right? But for us, when you are using Gator, what even though he has all these values, but what helps increase this more confirms that whether they're blockers or non-blockers are the Gator data. So here I just will present you a simple example where I took 3 samples from one plate position, and you can see that they're binding to, so 3 of these different plate samples, sample 1, 2, and 3, binding to antigen A. You can see that one of them doesn't even bind to antigen A, but 2 of them does bind. Now, when the same solutions, we are trying it with the receptor blocking, you can see in the next one that both of them block the receptor here, because after the receptor bound, there is no binding of these 2 supernatants here going on. But when we also do the epitope binning for these 2 particular sample, we see that, of course, after the biosimilar binds, one of them is not able to bind to the antigen anymore, but one of them bind. So even though just in this example of small, just a subset of 3, we find so much of diversity in information directly just by using a simple experiment, we can tell that among these 3, 1 is not even binding to the antigen. Now let's move on to the 2 that binds to the antigen. Of course, those are receptor blockers. And then when we compare the epitope with biosimilar, we see one of them has a similar epitope with the biosimilar and other one does not have. So in each plate, each of his 13 plates, we were able to find such diverse samples in good numbers that, at the end, we were having a hard time, like how to narrow down. So what we did was we worked ourselves and created a flow chart for him in order to understand all these experiments in one. So basically what we did was, first criteria is whether these samples that he gave us, does it bind to antigen A or its heterodimer, homodimer or heterodimer. Now, if they don't bind, of course, right away discarded those samples. But if they bind, then next thing we did is his next criteria was, do they block the receptors? So then we did the receptor blocking experiments. Then, of course, we found 2 subsets here where one blocked the receptor and one did not block the receptor. Then where we were able to even compare what are the off rates, and from there some of them were very tight binders. Now these receptor non-blockers, then we subjected them to biosimilar epitope binning. Among them, there were 2 categories. One were binding to the same epitope as the biosimilar, and the other were different epitopes than biosimilar. And same thing is the group that blocked the receptor also had 2 subgroups. So eventually we have narrowed for him 4 boxes just by doing this simple exercise where he gets to choose this tight binders from his periplasmic extract, that whether he has of course they all bind to the targets, but then do they block the receptor or they don't block the receptor? Are they in the same bin as the biosimilar, or they are in a different bin in the biosimilar or not? So that's how with Gator, if I have to summarize that how we have contributed more is first of all the throughput, like handling so much of samples in such short amount of time, because we were able to use the Gator Pro, our high throughput instrument. We were able to generate so much of data in a very short amount of time. And one good problem that we had, I would say, that the CAMOuse platform generates a whole lot of positive solutions. So for so much of positive solutions, how do you narrow down in lead selection? So then having multiple layers of information, not only off-rate ranking, but along with that, the receptor blocking and epitope binning, and cross-linking that data with the data obtained from his lab using ELISA and the FRET, that gave a unique combination for him and several layers for information regarding the single domain antibody selection for this particular project. So this is how, this is just one example how our users and are using Gator Bio because we have the throughput, we have the choices of biosensors, and you can design any kind of assay that suits. And that provides not only the given information that you have through the important platforms like ELISAs or FRED, but on top of that, you have additional information which helps you decide on your lead quite early on compared to later. So I'll end my talk here, and if there is any question.
Thank you, Libin and Indrani, for an excellent presentation. We received a few questions already, but we'll give the rest of you a moment to enter your questions in the Q&A box to the left of the slides. Before we begin the Q&A, I'll run through some brief announcements. First, I'd like to thank Benchling, Gator Bio, Sartorius, and WuXi Biologics for sponsoring this Digital Week. Next, I'd like to let you know that our flagship event, Antibody Engineering and Therapeutics US, is returning to San Diego this December. You can find more information about the agenda and lineup on our website, antibodyeng.com. What's more, there is still time to register for our Antibody Engineering and Therapeutics Europe event taking place in London, June 4th through 6th, and you can book your pass at antibodyengineeringeurope.com. Now back to our speakers to begin the Q&A. I'll begin with the first question. Can you elaborate why Gator is the next-gen BLI company?
Yes, the next-gen BLI company is, of course, as the BLI, is generally a package in terms of software, hardware, and biosensor, the three combination. And if you look into our, I can just, let's today talk about the biosensors in general. We have many choices that are new, which we launched, like we have anti-VH8, the Strep-Tactin XT, and also the entire portfolio of the AAV analytics. That is one thing. And it's not that we only have variety, but also the what we have done with the biosensors, we have pushed the detection limit in terms of higher end for AAV, if you think about AAV analytics, and even low sensitivity when you are thinking of nanobodies and antibodies. So this is just when we are thinking of biosensors. Along with that, our machines also, of course, add part, but I think there are many questions, so we can answer this more offline as well.
Yes, we can. Perfect. My next question, I think, is directed at you as well, Indrani. In SPR, there is a direct correlation between RU and the amount of mass bound on the surface with a distant formula. Is this also true with the NM shift in BLI?
Not just equivalent, I would say, because what happens is also in BLI, the refraction depends on the substance that we are capturing by the biosensors. That's why sometimes when it has a lot of water, the light just passes through it. So we might not be able to see anything even though it is captured. So it is not exactly proportional. There's a different formula. We can also send that formula to you offline.
Great, thank you. Next question for you. Is there any reason there are not many nanobody reagents on the market?
I can answer this question.
Okay, go ahead.
Yeah, so the reason is that, so before 2017, there's a patent that belonged to AVLinks. And so most of the effort is on the therapeutic field. And after that, after the expiration of the patent, and many institute and the company, they realized the potential of the single-domain antibody. So they started to using camel or llama or alpaca to generate single-domain antibody or nanobodies. I think right now the number is still much less compared to the monoclonal antibodies. However, you can see even from the publications, there are more and more nanobodies available from the market.
Thank you, Libin. Next question, is Nanobody good to be used for immunoassays such as ELISA, Western blot, immunostaining compared to mouse or rabbit hole antibodies?
Yes. So nanobody or heavy chain antibody, they are antibodies. So they have all the characteristics of the antibody. So they can bind to the antigen with very high affinity and also very specific. So basically they are antibody and they are just a small version of antibody. So all the immunoassays can use the single-domain antibodies to do that. And the benefit of using single-domain antibody is first, they are very easy to express, to manufacture. The second, you can generate multiple valent single-domain antibodies, so give you a unique feature that conventional antibody cannot have, such as you can make a bioepitope, and so they can bind even stronger to the antigen, so give you higher affinity or higher sensitivities. So there are many applications of the nanobody, not only in the immunoassay, but also in the therapeutic field, like bispecifics and also multiple functional biologics.
Great, thank you. And why do you select antibodies based on K-off only?
In this, I can answer this one. So in this particular case, because these are periplasmic solution, if it was not a periplasmic solution, it's just like a hybridoma supernatant, we can quantitate the amount of antibody. So we need to know the amount in order to understand the K-on, in order to obtain the K-on. Because if we know the formula for K-on, it needs concentration. But for this particular case, these are direct periplasmic solutions. So we cannot obtain that concentration. So then the other parameter what we can do is only K-off in this case. But this is just this particular case. You can do K-on, K-off, or KD also, if you have the concentration or if you know the concentration.
Great, thank you for answering that. Can you explain how blocking might affect the Gator binding data?
The blocking should not affect the Gator binding data. The reason is, first of all, in Gator, we don't need to block. What happens is in Gator is when anything that you are capturing the antigen in the biosensor only will bind to the antigen will only bind to the specific residues in the supernatant of these periplasmic solutions. So the interaction here is very specific, and we have wash steps which are automated in between that get rid of all these non-specific binding interaction. And sometimes even if you are not happy that you want to make sure there is no non-specific binding interaction, we do have just the media as a reference or several other reference where we do the background subtraction. And now even if you have something that you want to block the surface, you can come in and block, but it doesn't block the specific interaction. It only blocks if there is any non-specific interaction. So the blocking step is not needed for a Gator assay. And even if you want to do it, it just doesn't have an impact on it.
Great, thank you. My next question from the audience, can you elaborate on the differences between ELISA and Gator? And what would be an example of positive ELISA data but a negative Gator data?
Yes, I can explain this question. So actually, ELISA and Gator, and they use the same principle. Basically, is the antigen antibody binding. And however, the ELISA is different in some aspects different from the Gator. For example, in the ELISA, we usually coat the antigen on the surface. So sometimes we just put the antigen and in the coding buffer, and so the conformation sometimes may be hardened because of the coding procedure. However, in the Gator system, and we can use biotin conjugate the antigen. So in that case, the conformation or the epitope of the antigen can be exposed. So in more nature configurations. So that's kind of a little bit, that's kind of a difference from the conventional ELISA and the Gator. So I don't know whether Indrani has other more comments.
No. And also, it's more automated. You have less time and all. There are many other advantages as well.
Great, thank you. And how do you compare the nanobody generated from mouse to the one from camel?
So from the data so far we have, and the affinity and the specificity, they are very similar. Even we don't have a direct comparison, but based on the publication data, and I think the behavior are often the same. And also we have NGS data, and we compare the CDR3, the length of the CDR3, and we saw there's a extension in our camel mouse, and which is very similar to the camel, which has a longer or extended CDR3 regions. And so basically their behavior is very similar.
Great, thank you.
And
next question. Oh, go ahead.
Yeah. So one point I want to mention that in the camel or the llama, the heavy chain antibody, and then in the primary region 2, there's the four characterized amino acid. However, in the mouse, which single antibody, they are not exist in those in the primary region 2. However, they still behave very stable.
Great, thank you.
Yeah.
Next question. How did the Gator data help Dr. Xu for narrow down the leads?
Yeah, so because for the Phage displays, we can easily have thousands of banners. So from those banners, then we need a high throughput screening process for the early screening. So the Gator actually, especially the next generation BRI technology, they have high throughput. They put 32 probes at one time. So in a very short time, they can screening not only the binding but also the blocking activity. That's really powerful technology for early lead discovery.
Great, thank you. And I think we have time for another question. Are the nanobodies generated from CA mouse of human origin? Is additional humanization necessary for further use in clinical settings?
Yes. So as I mentioned that in the camel mouse, in this version, we use the mouse VH genes. So all the VH sequences come from the mouse itself. So for human therapeutic use, we have to humanize the VH genes, as camel or llama single antibodies. And also, our company also developed the fully human single antibody mouse. So stay tuned, and if you are interested, you can contact me.
Okay, great, perfect. And that's all the time we have for questions today. So thank you, Libin and Indrani, for a great session.
Thank you.
Thank you.
If anyone submitted a question that wasn't addressed, keep in mind that the speaker will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. Before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of Informa Connect Life Sciences, have a great day.